All right, let's talk about serotonin. You hear that word and your brain probably jumps straight to mood, right? Happiness, the so-called happy molecule. But what if I told you that's just scratching the surface? That serotonin has this whole secret life, one that's billions of years old and way more complicated, and frankly, way cooler than we ever imagined.
And this, right here, is the question that's going to turn everything on its head. I mean, think about it. Everything we're taught in biology class is that a messenger needs a receptor. It's like a key in a lock, right? But what if the key could just walk through the door? Because serotonin completely breaks that rule.
So, to really get what's going on, we're going to peel this back like an onion in 5 layers. We'll look at its dual identity, this ancient pathway it uses, how its receptors just love to break the rules, how they team up, and finally, why where a receptor is in a cell changes, well, everything.
Okay, first up, this idea that serotonin isn't just one kind of messenger. It's two. It's got the classic job we know about, knocking on the cell's front door to send a signal. But then it has this totally covert role, where it sneaks inside and starts rewiring things directly. And here's the clue that unlocks the whole mystery. This molecule is ancient. It was around for billions, with a B, of years before the whole receptor thing was even invented. And that can only mean one thing. It must have had an important job to do all by itself.
Now this slide lays it all out perfectly. On the left, you've got the textbook version we all learned. Serotonin bumps into a receptor on the outside of the cell, and bam, a signal gets sent inside. Simple. But now, look at what's happening on the right, inside the cell. Serotonin is physically attaching itself to other proteins, almost like a Lego brick. This process is called serotonylation, and it's a completely different ballgame. It's not about delivering a message, it's about becoming part of the machine.
So let's zoom in on this ancient pathway. This idea of serotonin acting as a direct molecular tag is, and I'm not exaggerating, a total game changer for how we should think about these molecules.
So what is serotonylation? Well, you can think of it like this. An enzyme called a transglutamase zips around inside the cell and literally glues a serotonin molecule onto another protein. It's like sticking a bright pink post-it note on it that says, "Do something different." And that serotonin tag completely changes what that protein does.
And just look at how widespread this is. It's not some weird, rare thing. We see it happening in our blood platelets to help them clot, in our muscle cells to help our blood vessels contract, in the pancreas to release insulin, even in our neurons, changing their actual physical shape. And maybe the most mind-blowing of all, it happens to histones, the proteins that literally package our DNA. That means serotonin can have a direct say in which of our genes get turned on and off.
So to sum that up, this one process, just sticking serotonin onto things, is essential for blood clotting, controlling your blood pressure, releasing insulin, physically reshaping your brain, and even controlling your genes. That is a staggering amount of influence for a mechanism most of us have never even heard of.
So we've got this super ancient, super powerful pathway that doesn't even need a receptor. But wait, it gets weirder. Because even when evolution did get around to building receptors for serotonin, they didn't always play by the rules either.
The classic model says a receptor is supposed to activate a go-between molecule called a G protein. That's signal transduction 101. But look at this. The 5-HT4 receptor can just go around it and use a different molecule called SRC to send its message. The 5-H2C receptor does the same thing with a partner called calmodulin. It's like they've all figured out their own private shortcuts to get the job done faster.
And if that wasn't weird enough for you, get this. Some serotonin receptors are just always on. They don't need serotonin at all to be active. They're just humming along, sending signals 24/7. And this suggests that something else must be controlling them.
And here's how that works. This shows the 5-HT6 receptor, which is one of those always-on types. And its job isn't determined by serotonin, but by which protein it's currently partnered up with on the inside of the cell. So when it's holding hands with a protein called CDK5, it kicks off the growth of new branches on a neuron. But then, when it switches partners to another protein called GPRN1, its job changes to extending those branches. Same receptor, totally different jobs, all depending on its internal dance partner.
And this idea of partnership brings us to our next level of complexity, because sometimes the most important relationship a receptor has is with another receptor.
This is such a cool way to see that principle in action. In panel A, you have the 5-H2A receptor just doing its own thing, sending its normal signal. But look at panel B. It is paired up with a totally different type of receptor, a glutamate receptor called MGLU2. And they've formed what's called a heterotrimer. Now, this isn't just two receptors standing next to each other. They've effectively fused into a single brand new signaling machine with completely new powers.
So let's break down what that new machine does. When the 5H2A receptor is flying solo, it uses a standard pathway called GQ. But when it's part of the team, that GQ signal gets way weaker. And get this, it hijacks its partner's ability to signal through a completely different pathway called GI. This is a total functional rewrite. And many scientists believe this exact mechanism is the key to why psychedelic drugs, which target this receptor, have such different effects than serotonin itself.
Which brings us to our final, and maybe most mind-bending layer of it all. It's not just what a receptor does or who it partners with. It's about where it is and when. It's real estate, basically. Location, location, location.
Okay, I know this diagram looks a little busy, but stick with me because it shows the beautiful choreography going on inside just one cell. Let's walk through it. Panel A shows us how signaling changes over time. Panels B and C show how a receptor's job is different depending on where it sits on the neuron. And panel D shows what happens when it's in a super specialized spot, a tiny cellular antenna called the primary cilium.
So let's break it down. First, panel A shows us that signaling is time-dependent. We just talked about this. The 5-HT6 receptor uses one partner to start building a new neuron branch, then switches to another to finish the job. Second, panels B and C show that a receptor on the neuron that sends the serotonin acts totally differently than the exact same receptor on the neuron that receives it. And third, and this is just amazing, panel D shows us that the 5-HT6 receptors in the main part of the cell are on, but identical ones way out on this little antenna are kept off by default, just waiting for a signal from a completely different receptor to flip their switch.
And that's really the ultimate takeaway here. A receptor isn't just a simple on-off switch. It's this dynamic, multi-talented tool whose entire job is defined by its context, where it is, who it's with, and what time it is.
You know, all of this perfectly illustrates this beautiful quote from biologist Francois Jacob. Evolution tinkers. This complexity we've been talking about, it isn't messy, it's brilliant, it's efficient. Evolution is the ultimate tinkerer. It took one simple ancient molecule and, through all these layers of regulation, gave it this unbelievable range of jobs to do. Which really just leaves us with one final thought. We came into this thinking of serotonin as the happy molecule, but we've seen it's also a construction worker, an electrician, a gene regulator, and a master collaborator. So if one single molecule that we thought we knew so well can be this incredibly complex, just imagine what other secrets are still hiding inside our own cells, just waiting for us to figure them out.